morning, everybody, and thank you, Mr. Chairperson. In fact, compared to presentations on science, this presentation on regulations can sometimes be very boring. So I'll see how best I can to make it interesting. But it is a very critical fact that in the translation of science into action or consumer benefit, effective regulation is extremely important. Now, as on date, we do not have any regulation on probiotics in India, and the regulator is the Food Safety and Standards Authority of India. The chairperson was here yesterday. But it's not because that efforts have not been made, because even the world over, the regulation is now sort of evolving as far as probiotics is concerned. But 2 years ago, the Indian Council of Medical Research set up a committee which was headed by Professor N.K. Ganguly. And many among us who are here in the audience were part of that particular committee, and we evolved what is called as the guidelines for probiotics. I was discussing yesterday with the chairperson that the Food Safety Authority should now adopt these guidelines and then develop these standards so that those who wish to market probiotics can approach the Food Safety Authority and get the approvals. The regulator actually acts like almost like a traffic cop, because as long as the policeman is there, everyone goes in order. The moment he's not there, is a total disorder. So this is true even to foods, and drug actually has a well-established regulation. I think my colleague will be speaking subsequently. But in food, there are still a lot of gray areas, and let us see how it evolves. Now, this ICMR DBT guidelines for evaluation of probiotics was published in the Indian Journal of Medical Research last year. So the status is there's no regulation or standard in India. Now, in India, we have two things. One is the Food Safety Authority regulates the food. The Bureau of Indian Standards lays down the standards for the industry to follow. Now, in the meantime, I very recently came across that the United States, there was a meeting of the Federal Regulation on Probiotics and analysis of the existing regulatory framework and the recommendations are made. This is the third meeting. They still have not come out with proper regulation. But some of the deliberations in this meeting were extremely interesting, and maybe we should just look into it. Even they have not adopted these. So what they have done is they have sort of categorized probiotic foods into different levels. Level 1 is called the basic level. This may not be a probiotic depending on the definition. That is, microorganisms used as starter cultures and preparations such as dahi. Of course, the word dahi was not there in the original document. The yogurt, I added this, whose role is to ferment a food and which then die in the stomach upon exposure to bile. So definition-wise, they are not probiotic, and this is often a question whether dahi is a probiotic or not. So this they considered is as a level 1 kind of a probiotic food where there is a microorganism which is responsible for the food to be made, but that microorganism does not have probiotic properties. Level 2 is safe but not clinically proven probiotic. We have quite a lot of them. People have been taking them, but no clinical evidence to show that they are probiotic come under level 2. That is probiotic which themselves have a long history of safe use but have not specifically been clinically tested, or the evidence is only based on increased numbers of the species in the stool. And these and all the strains at level 2 to 5. What happened? Yeah. These and all the strains and from level 2 to 5 must be genetically identifiable and meet the safety standards. Then level 3 is clinically documented probiotics. These are microorganisms whose utilization and delivery through various routes confer a basic effect that benefits the health and has been shown in human studies. Example, production of compounds that help create, restore or maintain homeostatic pH, improve the immunity and so on and so forth. And some of the probiotics about which we've been discussing the last couple of days would come under this. Level 4 is a specific probiotic health benefit. So these are microbial strains that have an additional specific function not conveyed by all other organisms and can be safely used. Here there is a very targeted outcome. You can actually measure the outcome. The earlier one was general well-being. These are probiotics with clear-cut benefit. It's quite possible that you could have probiotics both taking care of general well-being and also having evidence that they take care of certain specific health issues. And last, of course, is a narrow-use probiotic, microbial strains with very targeted usage. Example, if there is a strain which elevates certain neurochemical levels in the brain or something which is known to relieve stress, etc. This most often, I think, would be on the borderline between a food and a drug. And I would suggest probably this may go into drug. So these are the various levels which have been suggested, not adopted in the US, but these suggestions I think is worth considering so that we can have clear-cut categorization, avoid confusion in the minds of the people. So there is a comment of the working group members of the committee, is again interesting. The working group members noted that they also looked at the regulatory system in other countries and then said that the regulatory schemes for probiotic in countries like China and Japan are too culturally specific, that the regulatory mechanism in the EU are too restrictive, and those in India are untested. So, and then they also therefore said that if at all there is a country which has the capacity to bring about good probiotic regulation, probably it's the United States is in the position to provide guidance in this area, since they have the largest group of large probiotic market in their country. So I'm just quoting this because this has been a very recent information. Now let's go back to our guidelines. The guidelines deal with the use of probiotics in food only, and this does not refer to drug. It provides a requirement for assessment of safety and efficacy, and it provides a requirement for making health claims. Ultimately, what is the regulation looking at? First of all, the product which you are trying to market should be safe. And second, it should work. But you understand that the consumer is paying for the product with something which is promised on the label, and therefore that whatever is written on the label should be available to the consumer. That includes both efficacy and also in terms of the content of the microorganism, so on and so forth. So this is actually a flow chart, or what actually is the process which has to be taken place, and I'll go into details of each of these boxes. So demonstrate the specificity of the strain. So when you see somebody marketing saying this is a probiotic dahi, with no mention of what is the particular microorganism in that, such kind of product cannot be regulated, because you need to mention what is the strain, and that should be identified by PCR-based technique of 16S ribosomal RNA sequencing, or DNA fingerprinting techniques, or pulse field gel electrophoresis. The probiotic used in India should be deposited in an internationally recognized culture collection repository for traceability and for back reference. And then you need the following in vitro tests to say that the particular organism has probiotic capability. One, you should demonstrate in vitro that it resists gastric juice digestion, that it is resistant to bile acids, that the antimicrobial activity could exist against potential pathogenic bacteria, like by the production of bacteriocin, etc., and the ability to reduce pathogen adhesion to surfaces, and bile salt hydrolase activity. It is not essential that for approval everything has to be adhered to, but these are the steps which should be demonstrable and answers provided for that. Now come to in vivo safety studies in animal models. So we always wonder whether animal models are really relevant at all, because their gut is different, the food consumed by animals are different. But nevertheless, what is essential as per this is where we often and on go back to the Drug Act and look at the Schedule Y provisions. The entire formulation, here we are not talking about the safety of the bacteria alone, but you are talking about the entire formulation. So when you prepare a formulation, you will not only have bacteria, you will have many other ingredients, and therefore the entire product safety is therefore required. So there are three levels of safety. One is called acute safety, what's called a single dose or large dose. Here you are trying to see, as if you keep on increasing the dosage, does it cause an adverse effect at some level? Sometimes what happens, you may go even 100 times and you don't get an adverse effect. If you get an adverse effect, you call it as a low adverse effect level, or LOAEL, or and one level below the adverse effect level is called as the no observed adverse effect level. This is very a standard practice for any safety study. Identify whether adverse effect occurs at all. If so, you take the level below that, and that will be the safe level or upper limit of safety, which in turn should be at least 10 times or more than the normally consumed level. So you don't advise people to take at that no adverse effect level. You ask them to take it at a much lower level so that there's a huge margin of safety. Then we do what is called a subacute or a subchronic study, because since most of these probiotic containing foods will be something which will be taken on a daily basis. So you need to know that if I were to take a low dose or what we call as a normal acceptable daily intake dose on a long-term basis, does it have any cumulative effect? So this study could be of duration either 20 days or 90 days, depending upon what is the projected duration of usage. A lifelong, something used lifelong, then you go in for chronic toxicity study, it may even go up to 120 or 180 days. Even if you did not find any adverse effect in the acute study, we may still have to do these repeat dose, daily dose studies. So you then do an in vivo efficacy studies to substantiate the in vitro effect if you have suitable animal model. This is when we are making specifically, let us say, it improves immunity. Yesterday we heard an excellent presentation on the effect on immunity. Those are basically efficacy data. You take animal models and show that providing this particular probiotic food improves the immune function. Then evaluate on human use is to determine whether antibiotic resistant patterns, and it should be ascertained that any given probiotic strain is not a significant risk with regard to transferability of antibiotic resistance. Now, if there is a bacteria which is resistant to a particular antibiotic, is it possible that it will transfer that resistance from itself into the other gut bacteria? This is the same issue even when it comes to GM foods we are talking about, even though the probability of such a transfer is extremely remote. Now, if the strain under evaluation belongs to a species that is a known mammalian toxin, this is what happens is sometimes, let us say, as of date, we don't have that. Well, let's look at a Streptococcus species. We also know that some of the strains of Streptococci actually have toxic substances. They have streptolysis and so on. So if this probiotic bacteria is coming from a family where one of the members is known to have a pathogenic activity, you then have to demonstrate that this particular bacterium does not share those properties, nor does it have any kind of a genetic similarity to that. This is only for a remote possibility that some kind of a revertance can occur, and then a normal probiotic bacteria could suddenly turn into pathogenic. Now, then the assessment of the infectivity of a probiotic strain in immunocompromised individuals, this you cannot do as a planned study. This will come only as and when you get the information on the case studies. There are one or two instances of translocation of this bacteria in severely immunocompromised clinical situations, but they are very rare. But nevertheless, such data has to be provided to the regulator. If required, a label can be put on the product saying that not to be used for severely immunocompromised individuals if this particular fact is demonstrable. So then you evaluate the efficacy study. The principle or outcome of efficacy studies should be statistically and clinically significant in improving the condition. So you then have to have a very well-planned randomized double-blind placebo-controlled study, or at least a randomized open-label studies, and to show that whatever the clinical improvement you are expecting to put on your label is actually mitigated by this particular probiotic. And each of the parameters you study should have proven correlation with the probiotic tested. And probiotics delivered in food may not be tested in phase 3 studies, which is effectiveness studies, unless the product is making a specific health claim. So the kind of clinical studies one can do, of course, is to establish safety in a phase 1 type of a volunteer study, or in a limited number of individuals with any clinical condition you show an efficacy, which is your phase 2 kind of a study. A phase 3, which is a multicentric extensive studies where you look at the effectiveness, is not required, but if someone has done it in any other part of the world or is already being used in that data can be utilized here. So if a probiotic has a record of documented long and safe use outside the country, the data regarding this could be reviewed and deemed as sufficient. So whoever is applying for this need not redo all the studies. However, if you are making a health benefit claim of efficacy, then you need to do what is called the bridging study among Indians. It is quite likely that a health benefit which was demonstrable in a country like Japan may not actually happen if you do it in a country in India because of several differences in dietary habits, ethnic differences and so on. So you do a small study, you don't have to do an extensive study, using similar study designs but in a different population, and demonstrate that whatever effect is already being seen in other countries is also seen among the small Indian study, and that kind of a health claim can then be permitted on the label. So the effective dose of a probiotic is the minimal effective dose is the level of viable cells of the probiotic strain in terms of CFU in the carrier food that demonstrate general health promoting functions. So it is quite likely that you have demonstrated a health effect at a 10 to the power of 14 or 15, but in the probiotic food you are providing it only at 10 to the power of 5 or 6. Then it will not be meaningful because then you can't put the label because at that level you have not demonstrated the efficacy. So what is the minimal effective dose a consumer should take for him to get the health benefit should also be present on the label of the product. So the labeling requirements then are general labeling requirements under the food law, which is called the GSR 664 in our country, which tells that any food, what are the essential labeling, which includes of course manufacturing date, expiry date, best before dates and so on. And then in addition, you put the genus, species and strain designation following the standard international nomenclature. And the minimum viable number of each of those strains should be specified at the end of the shelf life. So when we do this, when the inspector looks at the product, he will take a product which is almost going to expire in a day or two and then calculate how many bacteria are there, and it should match up with what is put on the label. If it is more than that, there is no issue. Now, evidence-based health claims should be clearly stated. The suggested serving size to deliver the minimum effect of the product. This is again an important one, that you actually say that how much of the product has to be consumed, how often to get the desired effect. This will be entirely based on your details of your clinical studies. Very often we find that if you have to take 100 grams of something on a daily basis to get a health benefit, so sometimes people may just take 10 grams of it and expect the same benefit. It most often does not happen. So you have to therefore demonstrate how much one should consume to get that benefit. And then of course the proper storage conditions are to be mentioned. Then manufacturing and handling procedures are you must have adequate quality of assurance. They should all be done as per good manufacturing practices and follow the Codex General Principles of Food Hygiene or guidelines of HACCP and so on, and also use the guidelines of the BIS Good Hygiene Practices. So this is the overall gist of the entire process of regulation. Now the procedure as far as the Food Safety Authority is concerned is if one submits an application or a dossier seeking an approval, it then goes through a scientific panel. There are several scientific panels depending upon the kind of food you are producing, and I presume that the probiotics will go to the scientific panel on food supplements, nutraceuticals and so on, which is already functional. And the panel would deliberate, and then if there are any queries, they will get back to the applicant, and the applicant provides all the information. And once the panel approves, then it goes to the next level, which is called the scientific committee, which sort of endorses the views of the scientific panel after looking at a broad guideline and so on. And then it goes to an apex committee, and the apex committee is mostly an administrative committee, which generally is expected to give an approval for this particular product. We also have a committee on labeling and advertisements. So advertisement comes under the issue of labeling. Now there's a lot of controversy on advertisement. Many products are claiming things without actually having done clinical studies, but going to the media and making advertisement. So this particular scientific committee is now asked almost, not the committee, the authority is almost asked another 20 odd advertisers and manufacturers to give show evidence based on their clinical studies, whether the claim they are making in advertisement matches with the information they got out of the research study. So thank you very much for your kind attention.
Thank you. Thank you very much, Dr. Sasikaran, for excellent presentation and also raising issues relating to safety and efficacy studies and also on the legislative aspects, including labeling. And also, I think there are many issues that we could discuss. Since we are slightly running late, I would suggest that we could discuss at the end. But in the meantime, if there are any burning questions or clarifications you may like to seek from Dr. Sasikaran. Yes, please. So we'll take maybe two.
Thank you.
Related to the use of probiotics in immunocompromised individuals, and you talked about that. And now that there's a compelling evidence to indicate that the probiotics certainly improves the immunity. How about people? Is there any data, is there any hardcore data available on the use of probiotic in patients who are on immunosuppressive drugs? I'm talking of the transplanted individuals.
So as far as I know, there have not been any kind of a planned studies using these immunocompromised individuals, but wherever there have been case reports of some severe immunocompromised individuals, there has been evidence that these bacteria have translocated to other, not that they have caused any harm or something like that, that they translocate from there, and that probably is a risk. But planned studies on immunocompromised individuals, we don't have the data at all.
And the other point is patients who are taking immunosuppressive drugs. So
Yeah.
is there any
Naturally
interaction?
occurring immunosuppression itself, I don't think I have any data.
Can we take one or two more? Then we'll go to the second
Someone
speaker.
wanted a question. Dr. Mittal, you wanted to ask a question?
One
Can
more.
I ask one more
Yes, sir.
about the population? You said if the probiotic use has been proven in a particular ethnic group, say for example in the US population, which is very different from the Indian point of view, can that data be applied blindly in another population like in India, for example, where the genetic background, the food habits, everything changes?
Yes. So as far as the safety data is concerned, if it is being of safe use in other parts of the world, we accept them as such. When it comes to the efficacy data, and if it is specifically targeted at a particular ameliorating a disease component or something like that, then the bridging studies has to be done in India.
Professor Sasikaran, Professor Lahiri from Mumbai. I have a question as when we plan our clinical trials, as we need them real badly in India, when we go through suppose a phase 3 study, and we need to go through the ethical clearance which we have difficulties in getting it through because they wonder that there are no guidelines, there have been no approvals, formal approvals. In these situations, how do we go about doing a study?
I agree. This is a problem in the sense that if you are doing a study for a drug, you have got a mechanism which approves clinical trials. If you are doing for food, the food authority does not give approval for clinical trials. What you have to do is you get your ethical committee approval, you register it in the clinical trials registry of India and go ahead and do the study. And you only report to the food authority. As on date, that's a situation. Maybe in future there may be a situation that food authority also may adopt the same principle as the drug authority, but as on date, these two are required.
Okay, last two questions. One relating to that.
Sheila Krishnaswamy from Bangalore. Dr. Sasikaran here, behind the camera.
Yeah.
Go ahead, please.
You mentioned in one of the slides that for the labeling requirement, the number of servings has to be mentioned for a person to be able to take it for the right reason. But considering that it has a positive effect on so many different health problems, how does one decide on the number of servings or the minimum requirement for a person?
Actually, the serving size recommendation will come if you are making a generic kind of a thing, like it is good for your gut health or for immunity, etc. Then you just have to say that one serving is supposed to provide that. But if you have done a clinical studies where you have found that at a particular level you got the result, but below that you did not get any efficacy, then you have to mention that for you to get this desired health effect, you need to be at least 2 servings per day or 3 servings per day for is necessary. It's only when you are making a specific health claim where you already have data on how many servings has given the benefit. Otherwise, you can give a standard serving size and it's automatically assumed that 1 serving also will do you good.
Okay, last question.
Sir, I have 2-3 small questions. One, just transferable antibiotic resistance, is it okay only for one generation? Or is it to be shown that it does not transfer antibiotic resistance over generations of, because microbia divide very quickly? And that is one thing. Secondly, these other regulations, the 2 regulations, a non-requirement of large-scale multicentric studies in Indian population. I do not know why have we given it up, even if they have been done elsewhere. Because the bridging studies, whatever I have seen in the related fields of, say, vaccinology, have more been something like a joke, 30 children on this side or 30 children on that, which has hardly proven anything there in our Indian population. And so if this is the concept which we are extending to this also, then I think we need to think again.
Okay. As far as the antibiotic resistance is concerned, it is primarily based on historical data which you need to mention. There is no specific study which has been designed to demonstrate that antibiotic resistance. Now regarding the level of studies, now if the sponsor, or whoever is the applicant, wants to do a large scale study in the country, the food authority has no issue at all. But then what happens is quite often the other way around. Why should that not be a requirement? Is primarily because if you can demonstrate through your bridging studies in a population which is Indian, and that should be more often adequate. And we have actually taken some of these recommendations from the Codex kind of guidelines. The Codex is a guideline which takes care of the world over. So Codex has given that kind of bridging recommendation. And the multicentric studies primarily because of the investments involved and the large amount of infrastructure required, and may not give significant more information. But if a bridging study has shown a considerable amount of variation within that small sample size itself, then there may be a need for doing larger studies.
Thank you very much. Sorry we don't have enough time now. We'll have to move to the second speaker. Time permitting, maybe we come back. I think very, very important would be that while we have these guidelines from ICMR and DBT, but we should be discussing the way forward, what next. And in the area of research, perhaps it might be useful to develop generic protocols, and which can be then applied in multiple different centers. Otherwise, a lot of research being done, and not very sort of extremely useful. So with this, we move from food now to the drugs. And for this presentation, I'd like to introduce Dr. Hemant Koshia, who is the Commissioner, Gujarat State, Saurashtra University, and he has received over 9 fellowships from different organizations. Dr. Koshia, you have the floor, please.
Yeah. Good morning, friends. Respected Chair and participants. Sarve Sukhina Santu, Sarve Santu Niramya, Sarve Bhadrani Pashyantu, Ma Kashid Dukha Bhavet. Let all the people be happy. Let all the people be healthy. Let everybody feel bliss and comfort. And nobody should suffer from any misery. With this prayer, friends, there are stalwarts and scientists in field of the probiotics sitting in front of me. Dr. Talwar, Professor Nair, Dr. Mohan, an eminent speaker. Friends, I am not an expert in probiotic or in the subject. I am not a professor. I am a simple, a small regulator of one of the state of the India, that is from the Gujarat. Friend, Our main function is to look after the quality of the food and pharmaceuticals moving and produced within the border of the state and the country. As a scientist, it may not happen to you to connect directly with me because most of you have not come forward to get a license for food or drug to our offices across the India. But we have a indirect connection with all of you. We have a Drugs and Cosmetic Act. The morning you get up and brush your teeth, you are using that toothpaste. The quality of that paste, the licensing of that toothpaste is being done by my department. One step further, you go to the bath and using the toilet soap and shampoo. Again, it's cosmetics and quality monitoring and licensing is done by my department. If you go to the breakfast and have some probiotic food or some snacks, biscuit, cookie or tea, coffee, again, it's food articles. The quality of that food article and the licensing of that food article is done by the FDCA. If you fall sick and required to have a medicine, again, the licensing of that medicine and the quality monitoring of that medicine is done by my department and the team of the officer. So indirectly, we have a connection to one and all sitting in the audience. Friends, come to the main talk. In my talk, I will cover the regulation, environment, and present setup in the India. Because yesterday I have gone through all the sessions were very, very highly scientific. No speaker has touched the subject of this global and Indian market perspective. Are very, very important because you are working in the lab, but outside it's a huge market waiting for your product. So I will talk on this global market size. And again, of course, from the Gujarat where I am, and I will talk about working with my department and of the Gujarat. And probiotic, let food be thy medicine and medicine be thy food. The age-old quote from by Hippocrates. In India, what you claim, if you claim your probiotic as a food, you need to have a license under the Food Safety and Standard Act 2006 and the Rules and Regulation of 2011. If you claim as a dietary supplement in any form like capsule, tablet and powder, again, dietary supplement were recently included in the new Food Safety and Standard Act of 2006. And if you claim for any therapeutic on the product, it follows the definition of the drug and you require to have a drug license for your probiotic. So you have to be very cautious while labeling your product and marketing your product in India. Friend, this is Drugs and Cosmetic Act of 1940. Under this Drugs and Cosmetic Act, we are regulating the quality of the pharmaceuticals and the license of pharmaceuticals. And there are a lot of Schedule A to Schedule Y and a lot of chapters. Friends, if we look to the definition of the drug, all medicine for the internal or external use of human being or animal and all substances intended to be used for in the diagnosis. I'm giving the perfect word while which are more you have to be careful. That is diagnosis, treatment, mitigation or prevention of any disease or disorder in human being. If you claim your probiotic for these 4 points, that is the diagnosis, treatment, mitigation or prevention for any disease, then it attracts the definition of the drug and you have to have your product license under the Drugs and Cosmetic Act. So again, if you claim as a food, already Dr. B. Sasikaran has covered this food regulation, but I highlight the definition of the food under this Food Safety and Standard Act of 2006. Last year, on the 5th August 11, it was rolled out throughout the country. And under this act, we have a food safety and standard rules 2011, and we have a food safety and standard regulation 2011, where the food is given. These are the current regulatory scenario in India. As I mentioned, either it can be a food or it can be a drug based on your claim of your product. Friend, any legislation or any bureaucrat, because all the rules, regulation and acts are enacted for the well-being of the society, for the welfare of the society. But, in making legislation, India is not that proactive as compared to the developed nation. Once industry comes up, let some black sheep to enter through the string, then there is a lot of hue and cry, and government and parliament start working on to enactment of this act. So previously, as I said, the Drug Act was enacted in 1940. Then in those days, cosmetic was not covered under the Drug Act. In later part, some people had tried to adulteration in the cosmetics, and government has enacted the laws for the cosmetic, and it becomes Drugs and Cosmetic Act of 1940. Till 1960, even Ayurvedic medicines were not covered under the Drug and Cosmetic Act. Some Vaid and some Ayurvedic practitioner has started mixing some adulterants in the product, and government India has covered this Ayurvedic chapter also in this Drug and Cosmetic Act. And I hope, because probiotic, again, it's a very, very important segment, and yet industry is coming up, and I hope that it may be Drug, Cosmetic and Probiotic Act of India. Drugs are required to comply the standard as stated in the second schedule of the Drugs and Cosmetic Act of 1940. There are a schedule where the standards are mentioned. And this second schedule says if you are marketing your product probiotic as a drug, then you have to have prefix the pharmacopoeia if it is official in the pharmacopoeia. If it is not in Indian pharmacopoeia, you have to prefix the pharmacopoeia, whichever in the globe you have to, if it is official in the British pharmacopoeia or USP, then you have to, or Japanese pharmacopoeia, you have to label the product prefix with the IP, BP, USP or JP. And at all it is not in pharmacopoeia, then it goes as a patent and proprietary drugs. So drugs not included in the pharmacopoeia, it's like probiotic. Right now it's not under the pharmacopoeia, so it's a patent proprietary medicine, and the ingredients shall not interact with one another and do not affect the safety and therapeutic efficacy of the product. The combination shall not also lead to analytical difficulties for the purpose of assaying the content of the such ingredients separately. These are some salient features of this PNP medicine, that additives shall be innocuous, shall comply with the general requirement of the dosage form. If you are marketing your probiotic in a form of tablet, then again you have to comply the general requirement of that tablet, the dissolution time, destination time, the coating. That is common parameter of any tablet. If it is in a capsule form, then you have to comply the general parameter of the capsule and in addition to your product. This about all draft guideline, that is Dr. Sasikaran has spoke about this. Indian Council of Medical Research has come out with this draft guideline for the probiotic, especially in the food. These are main salient features of that draft guideline. If you want to market probiotic as a food, in India we have a Food Safety and Standard Authority of India. Yesterday we have with us Mr. K. Chandramouli. He is the chairperson of that authority, in the highest powerful authority of the India. Under this authority, there is a CEO, that is a Chief Operating Officer, Executive Officer, and there are offices of the state government. And FSA headquarters, there is a chief scientific officer. They have a chief scientific of in different categories. Some director for enforcement, director for training, like this. They have a general offices in different zones, and they have a commissioner of food safety in the State Department. Then with the commissioner of food safety, we have a designated officer and the licensing authority and the senior food safety officer and the food safety officers. Then we have a joint director of laboratories who take care of this quality control. That is the central food testing laboratories at the very regional level. And enforcement mechanism. Yet, because this food safety and standard authority of India were recently created, and they are setting this all mechanism and strengthening themselves. If you market your product as a drug, in India we have a DCGI, that is the Drug Control General of India, is the apex authority for approval of the drugs. Because as a state FDA commissioner, I am not authorized and not empowered under the Act to product license the new drug. Product license for the drug entering for the first time in the India. For those products, you have to approach the DCGI at Delhi. And if you are claiming your probiotic as a medicine, then if this strain is first time in India, then you have to go through the DC India and with all the safety efficacy data, and there is a committee, they go through it, examine it, and they issue the NOC to market your product in India. On the basis of NOC, any state regulator where your plant is located, you approach that state head, and that state head is issuing the license to manufacture that product. Once you are licensed from any state, you are free to sell this product throughout the country. Now, come to the organogram at the state level. This is the typical example of the Gujarat state. We have a commissioner, we have a joint commissioner, deputy commissioners. We have assistant commissioner in each and every district. We have a senior drug inspector, we have a senior food safety officer and food safety officer. These are the enforcement wing. We have a food and drug testing laboratories for this quality control. These are some important links if you want to learn more about this country's food safety and standard authority. That website is fsai.gov.in, and for drug it is CDSU, that is Central Drug Standard Control Organization, nic.in. Now come to the very important part of the market. If we look to the global probiotic industry's perspective, again, this global probiotic industry, it was $22 billion in 2010, and it's continuously growing over 14% in the globe. And if we look to the India, it's growing at the space of 25%. So in India, it is a very huge potential, because we are a population of 1.2 billion people, and because of there is a large pool of the young population, and people's paradigm shift is to make more health conscious and much more spending in this probiotic. Again, I said the India in 2010 it was a $27 million market, hardly 1% of the global market. But again, Indian market is growing at a space of 25% since last 5 years. Probiotic drugs account for 90% in India, because in India, a lot of pharmaceutical companies are marketing their product as a drug in Indian through the prescription and through the doctors. And thanks to the Yakult, they have entered in this probiotic market with the food category, and it's very rightly with the right claim. And in India, it's a probiotic market anticipated to reach 82 million by 2015. And these are the sector-wise food total, non-food total. This is sourced from ICMR ORG data. This Indian probiotic industry perspective, if you look, as I said, it's increase in the healthcare spending by the individual in the India. Pragmatic shift towards the preventive therapies rather than curative. We have a handful players, as Yakult is one of it, Amul, Nestle. Lesser competition and excellent growth opportunity in India. We have a proactive government support and minimum hindrance. I'm happy to give you the license either as a food or as a drug. You come to the Gujarat, and I'm happy to promote all this good probiotic food in the state of Gujarat. And if you look at the budget 2012 and 13, government India is also supporting this probiotic industry. Government priorities healthcare. Allocation of the fund of healthcare was hiked by 14% in this current fiscal budget, and the taxes on probiotic was reduced by 50%. Previously, the excise duty on the probiotic was 16%, and this year it is slashed by 50%. Now it is 8%. So government is also indirectly supporting this upcoming probiotic industry in India. Again, segment-wise market, huge in India, a lot of people, a lot of pharmaceutical company marketing as a drug as 90% and hardly 10% goes in the food sector. So again, these are some illustration list with the products of probiotic available in the market. This course, Yakult probiotic drinks, most of you are knowing and we have consumed while this conference also. We have a sporelec powder. It's again to treat the diarrhea, that is antibiotic-induced diarrhea. And sporelec, they're marketing this product as a drug since many years. We have some pro-life probiotic lassi. These are Vybec. There are some illustration as a marketing as a drug. Lactobacilli as a drug. These are some illustrative list. These are the product pre and probiotic capsules, Vybec. Because right now in drugs and cosmetic act we are lacking with the standard, but yet we have not come across this any complete of this product. And again, this standard is also very hardly. If somebody sends this product to test in my lab, though my lab is animal accredited, but want of standard, there's general examine if it is a capsule, they check for the general standard for the capsule, and they count the viable count, what is claimed on the label, and that should be remain there up to the last date of the expiry. These are some product pictures. These are all they are marketing pharmaceutical company in India as a drugs. And come to the place from where I'm here to share my views. It's Gujarat. It's situated in the western part of India. It's 5% of India's population, 6% of geographical area of the India. And every 2 years we are doing this biannual investor summit that is known as a Vibrant Gujarat. Last year we did in 2011, and where the partner countries were Japan and Canada. So this year I have Japan country again. They are a partner country, and I am also inviting this every participant to visit this Vibrant Summit, and I also invite Managing Director of this Yakult and his team to this forthcoming Vibrant. There is exhibition participants, 101 countries. There was 35,000 registered delegates last year, and over 1,400 foreign delegates has participated. There are few pictures of this event. This is of pharmaceutical investment comes through this every event. 2003 was only 14 billion, then 20, 152, 241, and last year it was closely 450, almost 10,000 crores of rupees. And come to the state of Gujarat. In Gujarat, there are 25 districts and provincials. We have a presence in each and every district, and headed by the Assistant Commissioner to take care of local licensing and the quality. If it is a food, the licensing is granted by that district head, and no need to travel all the way to the state capital Gandhinagar. We have a, as I said, it's an apex regulatory body in the state to grant the license of food, drugs, cosmetic and medical devices, and probiotic too. To regulate the manufacturing, distribution, and sale of this product, our main function is to issue the various certificate of WHO GMP, free sale certificates, non-coding certificate, and certificate of final product for the export purpose. Again, major functions to inspect the license units of this manufacturing and the sales, to draw the sample of the food and drug article moving across the border of the state of Gujarat, and to analyze that sample, initiate the complaint filed by the common man, and to prosecute if some wrongdoer is grossly violating the law and feeding the product as a counterfeit product in the court of law. So these are our main function as a state FDA, and thank you for patient hearing, and I am inviting all of you to the forthcoming Vibrant Gujarat Global Summit to happen on 11th and 12th of January 2013. Where again, this time also Japan is a country partner in this global summit. Thank you, friend. Thank you very much.
Thank you very much, Dr. Koshia, for a comprehensive presentation. And clearly you indicate that probiotic industry is growing at a very, very rapid rate as far as India is concerned. Yeah. And perhaps even more requirements for the role of legislation. And now I think we can open the floor for discussion. We could perhaps use about 15 minutes. And I was just thinking that what we could do is perhaps discuss the way forward. What next? We have the guidelines in the area of food, and also then there are systems in place as far as drugs are concerned. And if we can discuss what should be the guiding principles, and are existing mechanisms enough, or whether we need new mechanisms, and what steps further required in this particular area. So we start from here. Dr. Mohan.
Very nice presentation, sir. Thank you. One of the basic doubts I have as a clinician is that a lot of these are sold as drugs, and you showed some of them as capsules and so on. I understand that temperature, maintaining temperature in a refrigerator is extremely important. And yet I find many of the pharmacies which sell this don't tell this to patients, that you have to keep this in the refrigerator and so on. So these are quite expensive. Many of these are very expensive, and the patients take it, and I find them just keeping it in the house at room temperature. Won't they get destroyed? And isn't this temperature thing very important? And how long, even if you keep in a refrigerator, how long will they stay? Because they're supposed to be live microorganisms. So,
Sir, very important question. Because in Drugs and Cosmetic Act, we have a rules of Drugs and Cosmetic rules of 1945. Under these rules, we have a rule 65, which gives all the condition of the licensing for any retail pharmacy. Suppose you are a manufacturer of XYZ probiotic, and you are marketing this probiotic as a medicine, and you have put a storage condition that store below 25 degree, and accordingly you have calculated the shelf life of that product. Unless you store the product in the particular temperature, the shelf life may not remain same. So in India, as a regulator, we are facing a lot of problem with this cold chain, and this particularly in the retail market. In spite of best of the best factories, like Yakult, they have best their factories in place. They are following all of GMP norms, hookup standard, and everything. They produce the zero defect product. But once it goes out of their hand in the market, then the onus is on the retailer who is storing that product and waiting till the consumer come and take it away. So storage of the product is very, very vital and important. And if some chemist, but right now we don't have a clear guideline for the shop vendor, for the food business operator, for the chemist, we have a very guideline. If some erring chemist is not following the label condition of that particular drug to store in a refrigerator or air conditioner or particular temperature, then the as a district head, inspector, they are inspecting, they issuing the notices, sometimes they're cancelling the licensing, sometimes they're prosecuting that chemist. But it's a very, very valid question, and in India we are facing a lot of problem so far this storage condition is concerned, because in India we are diverse climate in northern part very cold extreme, in Kutch Bhuj of Gujarat 45 degree, in Rajasthan it is again 45 degree plus centigrade temperature. So one has to be very, very cautious and careful while storage and preserving of this probiotic up to the shelf life. Yes.
We actually do what's called as accelerated shelf life studies. In the accelerated shelf life studies, you can simulate the various conditions. So the regulator asks for that data. Now, if you say that within the storage condition, viability will be, let us say, for 6 months. If the storage conditions are suboptimal, what will be the viability? Now, based on that, the approval of the label also can be given.
Thank you very much. Yeah, over there. I think we can, I've been advised that we have only 5 more minutes. So we'll take only 3 questions, maybe short questions.
From the very beginning, could you please look at me?
Yeah.
Okay, please.
All right, thank you very much. Perhaps this question should be directed to the manufacturers, but as a commissioner, probably you can throw some light. From the past 2 lectures, I saw that if one is claiming it to be a food, the regulations are much less stringent than if it is given out as a drug. Yet I find from one of your slides that lots of companies are coming up probiotic as a drug. Why is it so? I mean, when something is more difficult to reach the market, yet this route is being chosen. Do you have any thoughts on that?
Sir, again, in India, we have a decentralized licensing system so far drug is concerned, even more decentralized in the food sector. Once you are licensed by one state drug regulator, then you are authorized to sell your product throughout the country. I have given all the illustration. None of the product is approved in the state FDA in Gujarat because they are not located within the border of the state of Gujarat. And for the, even this probiotic strain is well known. But if you market your product in a new dosage form, again it attracts as a definition of the new drug. If you come with the novel claim, again it attracts the definition of the new drug. If it is some innovation in your product, again it falls in the definition of new drug. And all the new drugs approved are legislatively to be given by the DCGI, that is central government. And in India, most of the states, maybe less infrastructure or less knowledge or competitiveness, they are issuing it left and right. So right now I think most of the companies marketing as a drug, but want of standard, and if it is a old product and without novel claim or no new dosage form, any state drug is issuing that product license, and again in pharmacopoeia there is no standard, and so I think in India, either way right now you are comfortable, but you have to select. Once you market your product as a medicine, then your outlet will be restricted to the chemist shop only. Once you are licensed into the food, because food selling, there are a lot of food business operator, more than 5.5 crores in India, as Mr. Chandra sir has said last day. And pharmacy is almost 5.5 lakhs across the India. So you are restricting your product to limited outlets, marketing as a drug. And again, to market your product as a drug, again, you cannot sell it directly. You have to go to the doctor. You have to generate your prescription, and then you can reach to the consumer. But once you are as a food, you can promote your product directly as you wish and as per your.
Okay, thank you very much. So last 2 questions, and then last yourself.
Combining probiotics with antibiotics.
Yes,
sir. You initially said that probiotics don't come under a certain drug category, so you have certain stringent criteria for those. But when you combine them with antibiotics, then how does it work? And there will be issues of drug resistance, whether these are resistant to the antibiotic with which these are being combined, etc. So how does one regulate that? And the second question is, I don't know, maybe to Dr. Sasikaran and also to you, the bridging studies. See, in vaccines, when we have bridging studies, we say efficacy, protection has been proven, you show immunogenicity. So what needs to be shown in the bridging studies in the case of probiotics?
sir.
See, basically, you already have demonstrated to large-scale phase 3 kind of studies in other country, and you have the data, which is published and peer-reviewed, and you're moving into another country. You don't have that kind of data in this country. So then you do the study, and the outcomes, whatever you measured in the other phase 3 study, the same you measured here. The only difference here is the duration of the study will be lesser, the sample size will be smaller. And then you demonstrate the same effect in this bridging study, then the authority accepts that even this population will behave exactly in the same way as the other populations.
Okay, last question, please. Sorry.
Sir, I'm addressing that combination of probiotic and the antibiotic. It's a common practice in India. A lot of pharmaceutical companies combining their probiotic, especially lactobacillus spore, with this amoxicillin or ampicillin. And many times, because as antibiotic, it's to be covered under drug license, they're adding the additional probiotic to make its value addition to that product to prevent the diarrhea mostly. And many times, sir, we come across, because of the poor coating of that spore they are putting inside with the antibiotic, and many times come down and not as per the label claim, and the product is not fitting the standard as per the label claim, and the promotion product are failing. And sometimes some manufacturers, to evade this DPCO and to come out the ambit of the price control. A very classic example I'm giving you, the doxycycline capsule. The government of India, the National Pharmaceutical Pricing Authority, has controlled the price of this doxycycline. So now pharmaceutical company has come with a smarter way, and they have stopped this doxycycline plain because it's not viable and profit margin is very, very thin. So now, to come out of the ambit of this price control, they're adding this spore microbes in that doxycycline. Now in India, virtually right now, plain doxycycline is vanished from the market, and all the doxycycline now available is with addition of this lactobacillus spore, and they are having a 4 or 5 times higher price, and then but it's a law of the land, and as a regulator, I have also taken a lot of this part with the NPPA to look into, and again have some new legislation on it. Thank you.
Okay, thank you. Last question.
I'm Dr. V.K. Batish. I'm President, I'm Secretary Probiotic Association of India. Actually, I have a very, discouraging kind of experience, regarding the reliability of probiotic products, particularly, when they are used as supplements. So we have done a lot of survey, we did a lot of screening of those samples. And to my great surprise, I find most of the capsules form of these products, they are falsely, labeled, and the exact, dosage is never done properly. The claimed effects, they are so variable from, say, 1 billion, we hardly get, say, a few thousands, many of the companies. I think 90% of those drugs. We did this work on maybe 5-6 years back. I don't know the present status, but I was really very sad. And secondly, the strain, even identity of the strain is also not done properly. Particularly Sporolac, I have a very, very, very different kind of impression. Sporolac, they claim that this is lactobacillus, but this turned out to be, just bacillus, we used some of the molecular techniques, we found that they were not connected with lactobacillus at all. So the situation is so alarming, and I feel that possibly because of the uncertainty in the regulatory standards of the country, so this situation is prevailing. And I'm telling you the truth that I'm receiving almost daily, a lot of requests from other countries to ask my opinion whether how they can get into the Indian market. So I'm not able to give them a good feedback because of this uncertainty in the. So I feel that this is the right time to take action now. Otherwise, a lot of spurious products now will enter the Indian market.
Sir, in beginning and opening remarks, as mentioned very clearly, any legislation, any act, any rules, it is for the well-being of the society, for the welfare of the people, and any legislation is formed looking to the common man's interest, the quality of the product we are manufacturing, and the genuine manufacturer of the genuine manufacturer. These are the main three parameters as a regulator we are looking in general. But sir, you have rightly mentioned, because right now there is no regulation. And as I am hopeful that this must be a drug, cosmetic and probiotic act looking forward. But unless and until there is a hue and cry from the society, there is a lot of representation, as you said right now to me. Because I am not the government, but I am a part of the state regulatory mechanism. But as a citizen, it's my concern too, because I'm also consuming a lot of probiotic food and pharmaceuticals and a lot of things. Even my father is studying 83 years of old, and he's also a doctor prescribed a lot of probiotic with antibiotic treatment. But right now I am a new spectator like you, because there is a want of legislation. And once, sir, whatever you said me, whatever you represent in this forum, as all as a active citizen, we should write to the government of India, we should write to the health ministry, and let the government to come out with the legislation to protect the interest of the genuine manufacturer, to protect the right kind of people who are judicially operating their units, and to weed out the black sheep from the trade and from the industries.
Okay, thank you very much. We have to now bring this session to the close. I'm sorry, many questions, but never enough time. I'd like to thank both the speakers for their excellent presentations and also for your interest in this session.
Sir, may I request?
Yeah, thank you.
So may I request you to please offer mementos to our speakers? To Dr. Shashikaran. ऐसे क्यों ला रहे हो? ओपन स्ट्रिप दैट. And now may I request Mr. Yoshiro Nakamura to please come on the dais and present the memento to Dr. Narayan. Before going on to our next session, again some housekeeping announcements. Regarding checkout, we have requested the hotel management that if they could, allow us to delay the checkout by 1.5 hours, that is by 1:30, we should check out. And for that, we could keep on continuing these sessions. At 1:30, once the sessions are over, you could go check out and then come and join us for lunch. That would be a good option for you all. And secondly is that reminding you again regarding the feedback forms, before leaving, please hand over the feedback forms. As I said, that is very helpful for our future programs. And also please collect your travel expenses. So now we go on to session 4. And I request Dr. K.R. Lahiri to come on the dais as a chairperson and to chair the session on shaping and modulating the gut microbiota composition. A few words on Dr. Lahiri. Professor Lahiri was former professor and head of pediatrics in KEM Hospital, Mumbai. She is currently the professor and head Department of Pediatrics in Dr. D.Y. Patel Medical College Hospital, Mumbai. She has 32 years of teaching experience. She is a fellow of the Indian Academy of Pediatrics, and she has received various awards and has over 250 publications in national and international peer-reviewed journals. Another commendable achievement of Professor Lahiri is that she was awarded for services during the Mumbai deluge by Mayor and Municipal Commissioner of Mumbai. Dr. Lahiri, please, if you would call the other speakers.
Thank you, Dr. Deepika, for those kind words. I would like to invite our two eminent speakers, Professor Satoshi and Professor Jean from France. I think we're running short on time. We should be, I think, precise and to the point regarding the time factor. I think the speakers, if you're not comfortable out here, you could step down and speak. That also could be okay. Because it's difficult to strain your neck and you'll get a wry neck before you reach home. So I think without much ado, we start off with the first speaker. Professor Satoshi is a senior researcher at the Yakult Central Institute for Microbiological Research. He has received the award of Japan Bifidus Foundation in 2003. He has over 32 publications and has reviewed in the reviewed journals and 4 book chapters. Professor Satoshi, you can start your lecture.
Thank you for your kind introduction, Lahiri. It is my pleasure to have the opportunity to give my talk in this symposium. I'd like to start my presentation.
Can you take the mic, please?
Sorry. Today I'd like to concentrate my talk about the relationship between the intestinal microbiota and colon cancer. As you know, the chronic inflammation is one of the risks of the several cancers, such as mesothelioma and hepatocellular carcinoma and gastric cancer. It's speculated that colon cancer may be regulated by the intestinal microbiota is responsible for the reason of the onset of the colon cancer. The most good explanation that chronic inflammation induced colon cancer is the case of ulcerative colitis. As you know, the risk of the colon cancer increase in the patients with long-standing IBD. The slide shows the direct evidence chronic inflammation induced colon cancer in murine model. As you know, the treatment of resistant sodium sulfate in this colitis in mice. We had observed the development of colitis-associated cancer, hereafter we call CSC, by treatment of 9th cycle of DSS. As shown here, histologically, CSC comprised proliferation of the gland epithelial cells, resulted in the formation of polypoid mass. We had previously described IL-6 trans-signaling, alternative IL-6 signaling promotes chronic colitis and CSC. As shown here, soluble form of IL-6 receptor plays an essential role on the generation of this signaling. Moreover, TNF-alpha converting enzyme, keratocase, play an important role of the generation of soluble IL-6 receptor by clearing the extracellular domain of membrane-bound IL-6 receptor. Released soluble IL-6 receptor induced IL-6 trans-signaling together with IL-6 and transmit the signal on the target cells. We had previously described the treatment of soluble GP130 Fc fusion protein, which was a competitive inhibitor of this signaling, inhibit the development of CSC. It is suggesting the trans signal is important for the induction of colitis-associated cancer. We had also described the lamina propria dendritic cells play an essential role in the generation of IL-6 signaling. We identified 2 distinct LPDC subsets in colitis mucosa by staining of the CD11B and CD11C. The first population is the CD11B high, CD11C high population as shown in here. The second population is the CD11B low, CD11C intermediate population. Both these subsets expressed cluster MHC and have APC activity. The former population expressed F480, the latter did not. So hereafter we call the former population as F480 positive LPDC, the latter population as F480 negative LPDC. As shown in here, F480 positive LPDC expressed taste and IS receptor. So we speculated that this population may be responsible for the generation of survival receptor. To confirm this possibility, we performed the next experiment. The slide shows the immunofluorescence analysis of the taste and IS receptor and IL-6 in both DC subsets. As shown in the slide, F8 positive LPDC co-expressed taste and IS receptor. On the other hand, F8 negative expressed neither taste nor IS6 receptor. Cultured F8 positive LPDC actively cleave survival receptor by stimulation of the PMA. That was a specific stimulator of the taste. On the other hand, F8 negative LPDC did not. On the contrary, IL-6 expression, both the protein and the messenger level in F8 negative LPDC is higher than that of F8 positive LPDC. So, since we made clear the essential role of each LPDC generate survival receptor and IL-6. Next question is how they are activated, especially focus on the activation of F8 positive LPDC. Here shows the cytokeratin immune staining of the CSC mucosa. You can see the presence of the erosion in the intestinal mucosa. Moreover, in this erosive region, you can see the deposit and intramucosal translocation of gut microbiota by fish analysis based on by using the probe recognize the consistent sequence of the bacteria 16S ribosomal DNA. In addition, some lamina propria mononucleus cells seem to eat gut microbiota. So we speculated that phagocytosis of the gut microbiota may be responsible for the activation of LPDC. Next, we confirm this possibility. The slide shows gives the staining of the F480 positive LPDC. As shown in the arrow, F480 positive LPDC 8 bacteria. To confirm, however, I don't show in the slide F480 negative LPDC never ate bacteria. To confirm this, we amplified bacterial 16S ribosomal DNA from both DC subsets. And we confirmed the amplification of the PCR product in F480 positive LPDC isolated from CSC. Next, we confirmed a kind of gut microbiota involved in the F480 positive LPDC by using the clone library analysis based on the bacterial 16S ribosomal DNA. Major clone in the F480 positive LPDC consisted Firmicutes and Bifidobacter. That was the major clone in the chronic content in the mice. So we speculated that phagocytosis of the gut microbiota in F480 positive LPDC may be responsible for the generation of survival receptor. To clarify this problem, we attempt to create FOH positive LPDC cell line from CSC. And we successfully established the FOH positive LPDC cell line IDEC5. IDEC5 expressed FO80 and class 2 MHC and TACE and IL-6 receptor. We don't show in the slide. They actively release solvite receptor by stimulation of TACE activators. Moreover, they eat microbe. By using this cell line, we confirm the effect of gut microbiome. By using this cell line, we examine the effect of 45 strain of human-derived gut microbiota on the activation of TACE. The results shown in this slide. TACE-inducing activity in the human gut microbiota classified into 3 groups. The first is the TACE inducer, as shown in the orange bar. The second is the TACE non-inducer, as shown in the blue bar. The resultant is the intermediate TACE inducer, as shown in the gray bar. Next, we confirm the effect of TACE inducer on the colon tumor genesis in mice model of colon cancer. TCR beta and P53 double mutant mice developed colon cancer in the presence of gut microbiota. These mice, importantly, these mice developed colon cancer by inoculation of human feces. So, this mice is a good model system to analyze the effect of human flora on the promotion of colon cancer. By using this beta decay of mice, we had created 2 kinds of notobiotic mice. The first is contaminated with 7 strain of TACE inducer. Another contaminated with the 7 strain of TACE non-inducer, and compare the tumor genes between 2 groups. The slide shows the cancer incidence in each notobiotic mice. TACE inducer actually promote the colon tumor in beta decay of mice. On the other hand, TACE non-inducer did not. Moreover, the colon cancer induced by the TACE inducer is inhibited by the treatment of soluble GP130 Fc fusion protein, which was a competitive inhibitor of IL-6 trans signaling. So, we can say TACE inducer actually induced the colon tumor by acceleration of IL-6 trans signaling. I'd like to switch gears to the next topics. Immunomodulating action is the typical biological function of Lactobacillus casei strain shirota, LCS. Immunomodulatory effect of LCS is 2-phased. The first is the immunostimulatory action. The second is the anti-inflammatory action. Anti-inflammatory action of LCS is confirmed not only in the animal studies such as type 1 diabetes and collagen-induced arthritis and lupus erythematosus and IBD, but also in human studies such as ulcerative colitis and norovirus-induced gastroenteritis and alcoholic liver inflammation. So, next we confirmed how LCS inhibits the inflammation. We focused on the effect of LCS on IL-6 synthesis in lamina propria mononuclear cells. The slide shows the method in brief. Laminar propria mononuclear cells, LPMC, were isolated from the mice-induced chronic colitis, and they were stimulated with LPS in the presence or absence of 4 strains of probiotic strain, including LCS. And the effect of LCS on IL-6 synthesis compared with other lactobacillus strain. The slide shows the result. The slide shows the comparison of the inhibitory effect of lactobacillus strain on IL-6 release. X-axis shows the inhibition of IL-6 release. As shown in the blue bar, LCS could inhibit the IL-6 production in LPS-stimulated LPMC in dose-dependent manner. On the other hand, the lactobacillus strain did not inhibit the IL-6 production. Next, we confirm what kind of cell components are involved in this anti-IL-6 action of LCS. We prepare the intact cell wall and protoplast and polysaccharide peptidoglycan complex PSPG from LCS and examine the anti-IL-6 action. Here shows the inhibitory action of the LCS and its component on IL-6 release. As shown in the previous slide, LCS could inhibit the IL-6 production in LPS-stimulated laminar propria mononuclear cells in dose-dependent fashion. Similarly, intact cell wall and PSPG fraction also inhibit the IL-6 production. However, protoplast fraction did not inhibit the IL-6 production. So we can say PSPG component of LCS is important for anti-IL-6 action of LCS. The slide shows the scheme of the cell wall structure of LCS. PSPG component of LCS are composed of 2 kinds of PSPG component. PSPG1, they are the large, over 300 kilodalton, and small PSPG2, they are the 3 kilodalton. Next, we prepared both the PSPG component and examine the anti-IL-6 action. The slides show the result. As you can see, the PSPG1 inhibits the IL-6 production. However, PSPG2 did not inhibit the IL-6 production. To confirm this possibility, we created a mutant strain which lacked PSPG1 by gene engineering method. The slide shows the gel filtration of the polysaccharide peptidoglycan complex in LCS and the mutant LCS. As shown in the previous slide, PSPG component of LCS are consisted of PSPG1 and PSPG2. However, the mutant strain actually lack PSPG1, while the PSPG2 components are intact. Then we prepare the anti-IL-6 action between 2 groups. The slide shows the inhibition of IL-6 production and PSPG1 expression of LCS and mutant LCS, which judged by the flow cytometer by using the monoclonal antibodies against PSPG1. LCS and control mutant strain, which have the intact PSPG1, can inhibit the IL-6 action. On the other hand, PSPG1 mutant strain, which lack intact PSPG1 did not inhibit the IL-6 action. So we can say the PSPG1 component actually involved in the anti-IL-6 action of LCS. Finally, we carried out the in vivo effect of LCS in the colon cancer model. The slide shows the experimental method in brief. The mice were divided into 3 groups and given LCS or mutant LCS strain and saline during the induction of CSE. Finally, the cancer incidence were compared among 3 groups. The bottom of the graph shows the recovery of LCS and mutant LCS from the feces from the mice. The recovery rate comparable between the LCS and mutant LCS. The slide shows the cancer incidence. As shown in the slide, treatment with LCS actually inhibits the development of colon cancer. However, interestingly, mutant strain did not inhibit the colon cancer. Moreover, the inhibition of the colon cancer by the treatment of LCS is associated with the downregulation of IL-6 and SOC3, which was a negative regulator of IL-6. So we can say LCS inhibits the colon cancer by downregulation of IL-6 transsignaling. The slide summarizes today's my talk. Under the inflammatory condition, intestinal microbiota induced IL-6 transsignaling on the colonic epithelial cells. And details mechanisms unclear. We speculated the excess amount of IL-6 transsignaling moderate colon tumor genesis. And LCS inhibit this signaling pathway is the mechanism involved in the anti-inflammatory action of LCS. I'd like to thank the current and former member of the cancer research group in the Yakult Central Institute, and Dr. Mitsuyama and Tsuruta for his valuable advice in this experiment. Dr. Jagan Shura and Stefan Lozion, they were provided as a soluble GP130 Fc fusion protein. Thank you very much. Diane Ward.
Thank you very much.
That's your brain.
For that excellent piece of work which you have shown about the trans-signaling of IL-6. And it's quite scary that a simple thing like inflammatory bowel disease, we think it's not very tumorogenic, can be tumorogenic, and the role of LCS in preventing and treating the complications maybe. The floor is open for questions. I would like some comments from experts from Tata Memorial, if they have any experiences. Professor Mohandas, Professor Shubhada, if she's in the audience. Yes, sure. Give her the mic, please.
Wonderful lecture. Congratulations. My question is regarding what happens to the other members of the IL-6 family, like oncostatin M1, or would they take over the function?
I did check the other member of the IL-6 family. Sorry.
Any questions from the postgraduates and the youngsters? We don't see hands from behind. I think you should not shy away, and if you have any queries, please ask. Any other questions for Professor Satoshi? Yes, please. Yes.
There was a very, very nice talk.
Thank you very much.
I just wanted to understand, have you also looked at the role of T-cell growth factor? When you're looking at the immunosuppressive cytokines like IL-6, whether or not the T-cell growth factor, because that has been shown conclusively in many situations that dendritic cells which are immature are able to induce a very high levels of immunosuppressive cytokines.
So I can answer the question. What is the point of your question?
My point of view is that if you're looking at the inhibition of lactobacillus strain-induced inhibition of IL-6, have you also looked into the T-cell growth factor, the TGF-beta, for example?
I don't check the TGF-beta production is not inhibited by LCS. I check. TNF-alpha or IL-6 is a major component of the target of LCS.
All right.
Any other questions? Thank you, Professor Satoshi, for that excellent talk.
Thank you very much.
We move on to the second speaker. Give a round of applause, please. Dr. Professor Jean-Antoine from France. Bonjour. You're welcome to this August gathering. He is the Expert Nutrition Director, Danone Research, France. He has received fellowship in ILSI Europe Nutrition Leadership Program, and has also received gold medal in medicine and silver medal for his work. He has over 84 publications in peer-reviewed journals and 8 book chapters. We know that our bodies are living ecosystems, and by the time we are few hours old, they have invaded us in a big way. And this crosstalk between immune cells and microbes, which Professor Jean is going to dilate upon today, plays a very crucial role. Let's see whether it's a civil conversation or is it an argument. You can start, sir.
I was impressed yesterday when I was attending the cocktail, and we were within the same species, but using different languages to exchange ideas and concepts. And I was wondering whether or not microbes can do the same things, discussing with different languages and exchange ideas or concepts. And this morning I also feel like a kind of probiotic, because I am from the same species, but I am a foreigner, and I will spend some time in a new place, Bangalore, and trying to use my own language, a kind of English, to share with you some examples and to try to change your mind and your knowledge. And in fact, I'm coming from France, in Paris, near Paris, in our research center, and our company ambition is to bring health through food to as many people as possible. And we do believe that food can have an impact on health. And that's a challenge for all of us. Because one of the big challenges is to use the food we have, including some of the dairy food and the yogurt which are there, to fulfill our dietary needs. And in between, as you know, we have this strange organ which is the gut. And we have the responsibility to manage the adequacy of our food intake with our dietary requirements. And in fact, we are all autonomous eaters, and sometimes overeaters, because we are the only ones who are able to manage our intake, because we are eating in different places. And it's up to us to find the right balance of our food to match our dietary needs. And the second big challenge, as Joël Doré was telling you yesterday, is that we are full of microbes. And in fact, we have to look at us as a supra-organism where microbes are the real factor. And when I look at you, I start to look more at microbes than human beings. And in fact, just to give you some figures and to remember you some figures, that our gut microbiota is roughly 2 kilograms. And if you look at that we are shadowing behind the microbes. And more impressive, if you look at the gene, the size of the gene, that's our human genes, and that's the genes of the microbiota. So in fact, our real genome is in the microbiota. Which means, by the way, that our genome, the microbiota genome, can change. And Joel told you yesterday that depending on your culture, on your diet, your gene will change. So we have to realize that our genomes are changing. And another concept that we have to share and to understand that these microbes are a huge diversity. In our microbiota, we have 6 phyla. What is a phylum? Well, if you look at the kingdom of animals, we have 14 phyla, and one of the phyla is the chordate. And the phylum is going from snake to bear. And that's a phylum. And in the gut, we have 6 phyla. And if you look at this animal, you have to go back to the genus there and to the species. As human beings, we are one species, only one species. As microbes, we have 6 phyla. So we have to understand and to realize that we are inside a huge diversity. I am always wondering whether or not there is the same language between the snake and the bear, because they have to exchange some things. And more challenging if we can have the same language between sponge, for example, bear or spider. If we look at the gut microbiota, we have different phyla over there. And if you look to the genus, we are speaking about genus, we are at the end of the spectrum. How can a few probiotics can use the same language to speak to this huge diversity of microorganisms? Just to remember you, because yesterday there were mentioned that we have no simple definition of probiotics. We have one, and the WHO gives that one, which is agreed by everyone that, probiotics are live microorganisms which, when administered in adequate amounts, confer a health benefit on the host. Where are they on the distribution? Well, we have some of them which are there in that phylum. Some of those bifidobacteria in another phylum. And even outside of the domain of microorganisms, we have some yeast, like Saccharomyces or kefir. And we are dealing with this concept that these different microorganisms can speak to the gut microbiota. And even if we are speaking with the same genus, we have different species, and within the same species, we have different strains. So we have to go to the one strain, to the huge diversity of the gut microbiota. And to make you understand that, well, I use the example of dogs. Dogs are within the same genus, but depending on the species, you can have different capacities. And you can select the capacity you want to achieve the job you want. But even there, if you go to the strain, you can see that one dog is able to search for drugs, but the other one is looking for the gut microbiota of the other dog. So depending on the training, you won't have the same effect. So all the dogs are sharing the same basic capacities, but they are similar, they are not identical, and they may have some specific capacities. Probiotics are also specific, species and strain specific. So we can, in a way, speak about probiotic at large. But when we are dealing with probiotic, we have to look at the species and the strain, and we cannot extrapolate from one strain to another strain. Which means that with two different strains of Lactobacillus casei, and Shirota is one example, we can't extrapolate to another strain to expect the same benefit. So can we have a crosstalk? First, if you want to talk, you need to be living. But it was said that we have in our digestive tract a gate which is the stomach, very acidic, and it's a drastic gate, and it is supposed to kill most of the microbes. In fact, if you use different strains of lactobacilli, which is one part of the yogurt, So if you expose them to a stress, a biliary stress or the acidic stress, so we start with a concentration of 10 to the 8 in a tube. You wait for 1 hour during the stress, and you can see that depending on the strain, they are more or less sensitive to the biliary stress or the acidic stress. And in the gut, you have to support, to bear the acidic stress first and then the biliary stress. And if you look at that, those who are able to resist to the biliary stress are sensitive to the acidic stress. And you have a few strains which are able to support both stresses. Which means that some yogurt can survive in the gut, but some yogurt will not survive in the gut. Second, well, if you use another strain, another species, which is a Bifidobacterium, and that's a test we have done in humans, when people are ingesting 10 to the 8 Bifidobacteria, if you look at the concentration of Bifidobacteria in the stomach with a tube you put in the stomach, you can see that for 1 hour, the concentration of Bifidobacteria ingested as a fermented milk can resist, and we have still 10 to the 8 Bifidobacteria in the stomach after 1 hour. If you use another strain, the product which was on the market, that strain was sensitive and was destroyed by the acidic condition. Which means that if you want to say something, you have to test the efficacy. And if you want to say that your strain of probiotic is able to survive, you have to test it in human beings. To survive in the stomach is good, but then you have to survive in the small intestine. So using the same strain of Bifidobacteria, we look at the concentration of Bifidobacteria in the small intestine, that's the control meal without Bifidobacteria. And you can see that after 1 hour, you can see 10 to the 8 Bifidobacteria entering the small intestine. And during 4 to 6 hours, you have a concentration of 10 to the 8 Bifidobacteria in the small intestine. Meaning that some probiotics can survive in the stomach and in the small intestine. Same thing for another probiotics. We are using another LKZI. Again, after 1 hour, you can see 10 to the 8 LKZI in the small intestine, and for 2 to 6 hours, which is the time of transit in the small intestine, you can have this concentration of 10 to the 8, like specific Lactobacillus KZI. Well, going in through the digestive tract, you have to survive in the stomach, then to go through the small intestine, where there are a few microbes, but then you have to enter the huge colon where there is a huge crowd of microorganisms. And you have to survive that if you are a good probiotics. So look in the stool, and before ingestion, that was done on 5 volunteers, before ingestion, there are some Bifidobacteria in the stool, some endogenous Bifidobacteria. Before and after ingestion, you have the same concentration. Before ingestion, we don't have our specific probiotic in the stool, which is normal because it has not been ingested. After 1 week of ingestion, you can see that we have been able to find 10 to the 8 to 10 to the 9 Bifidobacteria per gram of stools, demonstrating that some probiotics are able to go through the human digestive tract and survive at 10 to the 8, which was the concentration ingested during the meal. And if we look at the kinetics, which is quite important, so looking before ingesting the probiotic, during the ingestion of probiotic, and after ingestion of probiotics, when you don't ingest any probiotics, any Bifidobacteria, there is no change in the concentration in the stool. But when you eat a specific probiotics, before, during, you can see the increase. But what is important there is that when you stop eating the probiotic will disappear. And to control that, we have a marker, I0 marker. Before, there is nothing. During the ingestion of the marker, you have the stable concentration of the marker. And when you stop ingesting the marker, there is a decrease in the marker. You can see that we have the same slope of decrease between the probiotic and the marker, which means that probiotics are transient. I will be there for 2 days, and then I will be going elsewhere. Same thing for another probiotic, which is the LKZI, before, during, and after ingestion. And within 1 week, roughly, all probiotics are gone. So some strain can survive transiting through the human gut, which is good. But can they be active? It's good to transit, but you have to be active. To do that, we use an animal model, hypnotoxenic rats in INRA, near our research center in Palaiseau. And they inserted close to the lactose operand a luciferase gene, which means that when the gene is not active, you are not activating the luciferase gene, so you are not producing light. But when the strain is active and using lactose to produce energy, you are stimulating the luciferase gene and you switch on the light, which is fantastic because then you have microbes in the dark of the gut and you can see the light when they are working. And we have done that in this animal with the human gut flora. And you can see that in the small intestine, after 1 hour and a half, when the microbes are entering in the small intestine, the light were switched on. Saying, in fact, the microbe was there and were able to work. And they were able to work for a few hours, but they were more active for a longer time in the colon, where they are staying for a longer time. And you can see that after 1 or 2 hours, the probiotics are there, living and active. Well, they are active, but okay, can they change the gut microbiota? That's a study that has been done in kids, young kids, 1 year and a half, before, during, and after ingestion of different products, a control milk, a yogurt, and a yogurt with a specific probiotic LTZI, and we look at the number of kids with an adequate concentration of lactobacilli in their stool. You can see that in the control group, 20% only of the kids had enough lactobacilli in their stools. When they have yogurt, you can see that we were able to observe an increase in the number of kids with adequate number of lactobacilli. When they used the specific probiotics, we had a huge increase. Roughly 60% of the kids had enough lactobacilli. So a probiotics was able to increase, to improve the concentration of good bacteria in the gut of these kids. Again, when you stop ingesting the probiotic, you will lose the effect within 1 or 2 weeks. Doing that in adults, so we look at the map of the microflora in adults using a specific apparatus that has been developed by Yakult, the Flora Scan, where you have different concentration of different components of the microbiota. So when you ingest a control yogurt, you have this picture. When you ingest a specific yogurt with a specific probiotic, you have another picture. And obviously you have to compare both, and you can see that ingesting a probiotic, first you are able to find the specific components, so your probiotic is surviving, so you find it in the stool, the lactobacilli which are going along with the bifidobacteria, but you are able to see that there are different increase in the composition of the gut microbiota. So probiotics are passing by and they are changing the composition of the gut microbiota. It's good to change the composition, but what is more important, and you remember again the message from Joël, to know who is there is important, to know what you are able to do is more important. So can they change the function? Well, you use another model with Jeffrey Gordon in St. Louis, in his lab, where they colonize mice with different probiotics. And in fact, they look at the gene of the microbes of the gut. And the major microbes in these mice were Bacteroides, Theta Omicron, which is a specific species. So you have a lot of genes which are there, and when mice are ingesting a specific probiotic, you can see that some of the genes of the microbiota are switched on. So a microbe is passing by and change the genes which are functioning in the gut microbiota. And if you use another probiotic, you can see that this other probiotic, ALTCI, will turn on some different gene as the bifidus. So without changing the composition, you change the function of the gut microbiota. And you can see that the genes are changed. Well, is it an efficient crosstalk? Well, the two models, the classical model is the effect on diarrhea. So we look at the efficacy of probiotic on children diarrhea. So we recruited roughly 900 of kids, and we asked them to use the probiotic for 12 weeks, and we look after that. And we look at the incidence of diarrhea, and you can see that using a specific probiotic, you can reduce the incidence of diarrhea. So the probiotic is there, passing by, and telling the gut microbiota to fight better against, or to recover quicker from diarrhea. And we have done a study in India some 10 years ago, using 75 children hospitalized for common diarrhea or being in daycare facilities, and they were receiving the WHO formula, plus either a controlled UHT milk, or local fermented milk, the dahi, or a milk with a specific probiotic LKZI. And as the result, so the control groups, they were staying in the hospital 2 days roughly. The local dahi has a small effect, but you can see that the specific probiotic was able to reduce or to induce a faster recovery in these kids hospitalized for diarrhea. So we can have an effect restoring the recovery or restoring the capacity of the gut microbiota to recover from diarrhea. To demonstrate the efficacy to restore the barrier, we use a model where physicians are destroying the barrier, and the best way to damage the barrier is to use antibiotics. So that study was done in UK, where they used 135 elderly, which were hospitalized, and they received different antibiotics, either orally or parenterally. And within 24 hours of the start of the antibiotics, they were receiving either a non-fermented dairy food or a specific probiotic, LKZI. And this probiotic was given during the antibiotic treatment plus 7 days after the antibiotic treatment was stopped. And the criteria was the number of diarrhea and the incidence of Clostridium difficile in the stools. And the results are the following. In the control group in yellow, you can see that the number of diarrhea is reduced. So the number of diarrhea is reduced between the control and the probiotic. But more important, the number of the diarrhea that was induced by Clostridium difficile was suppressed by the probiotic. And we have to understand that when you damage or destroy the barrier, well, you induce the risk of diarrhea. When you restore the barrier, you reduce the risk of diarrhea, and you can prevent the occurrence of the opportunistic pathogens. So, ingestion of some probiotics can change the gut microbiota composition and the functions. So we were looking at the efficacy of probiotics on the microbiota, but are they able to work on our cells? Can we imagine that some microbes are speaking to our cells? And using the same models, we look at the gene in rats of the intestinal cells when the rats were in the normal condition, in germ-free condition, or when they were ingesting LKZI or Bifidus animalis. And surprisingly, when they were ingesting LKZI, the gene of the cells were switched on or off. So the ingestion of a probiotic can change the function of our cells. And if you use another probiotic, you can see that there are some specific genes which are turned on by LKZI or by another probiotic, but we have also some commonalities. So we have differences between the probiotics, but we have also some commonalities. Well, and for me, it was a kind of revolution. A microbe is passing by and say, well, hey, gut, change your functions. What do we mean, change the function? Well, we look at the mucus composition. And to do that, we use some intestinal cell, HT29, producing the mucus. And we cultivate the cells, and we infected them with rotavirus, which is able to stick to the mucus. And using antibody, you can see that the rotavirus is able to stick to the mucus of the cells. When you cultivate the cells with a specific LKZI, the probiotic, and then you infest them with the rotavirus is no longer able to stick to the mucus. So we look at the composition of the, we look at the composition of the mucus, and in fact, the incubation with LKZI was able to change the composition of the mucus, which means that the probiotic was passing by and telling the cell, change the composition of the mucus, and the cell obeyed. And if you change the composition of the mucus, then the rotavirus is no longer able to stick to the mucus and you reduce the infection. Which is for me a good example where we can understand the role of probiotics. They are not fighting against the virus by themselves, but they are helping the body, or they are helping us to adapt to the challenge of the rotavirus. They are collaborating with us to improve our function, our defense functions. So ingestion of some probiotic can change the gene expression of the cell of the human gut. So we are really dependent on our microbiota. We are really dependent on our diet and ingestion of probiotics. That was done in animals, but recently it has been done in humans. So people have put a tube in the small intestine and take some sample of the epithelium and look at the gene of the epithelium with different probiotics. And there, in human beings, it has been demonstrated that you can switch on or off genes of the cell of the upper part of the small intestine. So we are really at the edge of a new discovery where we have to understand that our gut functions depending on the microbiota which is there. So to summarize, first, remember that we are a superorganisms and we are mainly microbes. And we are achieving a lot of different functions, but again, with age, our gut microbiota is changing. Our genes are changing with age and depending on our diet too. We have to forget the stability of our genome capital. Probiotics, they are able to speak with the gut microbiota, and the gut microbiota will change the message for the gut cells, or even and change the function of the gut cells. And it was mentioned yesterday that we have a lot of different functions, the endocrine function, or the defense function, or the immune functions. But also the probiotic can maybe act directly on the gut cells. So 1 century ago, Mechnikov was saying, hey, when we will understand how to improve the gut microbiota, maybe we will be able to live beyond 70 years, and I hope to do so. And right now, we have this new quote for Jeffrey Gordon saying that we have to take into account that we are living with microorganisms outside, but also inside, and that's a new, totally new physiology we have to learn from. Still, a big question is there. I am using a kind of English. Which languages are using these microbes? And are probiotics able to speak to all of the components of the gut microbiota? Question mark, I don't know. Some people are listening. Bacteroides is listening to Ehrlichia and Bifidus, but are all phyla listening to one microbe? I don't know. So is the message heard by all the microbes or not? I don't know. Are they using this old tricks thing, speaking to one, or are they touching their cells? And we have the TLR receptor, so what kind of language are they using? I don't know. Are they using the Twitter? I don't know if you have the answer, but I am ready to listen to your question. Thank you.
Thank you, Professor John. Thanks for reinforcing our ideas about the crosstalk. We know it all, but there are gene expression findings which are really interesting. And I think everything starts in utero with the baby and the mother. And of course the gynecologists and the pediatricians working in tandem, which is so very important. As you mentioned, diarrhea, antibiotics being used unnecessarily for children, where the pioneer bacteria is destroyed forever. So I think these are a few points which are very, very important. With that, I think we could have a few questions. Yes, Professor.
Sees that altering the expression is occurring at the gene methylation level in the crypt cells, or is it occurring at a post-translation level in the mature cells?
Well, the gene were observed. It was the epithelial cells, typically. And they are working right now to switch the gene, and we are monitoring the transcription genes. So we are really interacting with the gene expression. Right now, we have to go maybe further to go to the proteomics and elsewhere. But the big message for me is that, well, they are reacting. How far? It's an open question.
Any other questions? Yes?
A very interesting lecture. I'd like to know what is the effect of probiotics on cell senescence? Like, do they directly affect telomerases or DNA repair ability of these cells?
Well, I don't know. It's a field open for research, but what they are doing right now is to annotate the gene. So you could do it both ways. So either you look at the gene, you are able to change, or you look at the effect on the clinical or functional levels. And then you have to match to see that. But, well, I don't know what are the mechanisms. Again, I don't know what is the language and what are the mechanisms. Are they using the same language for the same mechanism? Again, it's an open question. What we are observing and reporting that there is a dialogue. Well, there is a dialogue. They are able to cross talk, and talk is in brackets.
Yeah, that's one aspect where they're cross-talking with the gut microbiota, but it would be also interesting to understand how they talk directly with the cells. The intestinal, how they directly talk with the gut cells. That is why whether the repair capacity of the cells or their aging, the telomerase is etched, anything changing in them.
One of the ideas is that first they are producing short-chain fatty acid, for example, but second also there are some cells, some microbes which are able to stick to the cells, because in the small intestine you know that there is not a lot of mucus, so there is direct contact between probiotics and the cells, so that's why I was mentioning this touching with referring to the TLR. Again, it's a new physiology. What I am trying to tell you is, hey, it's an open field. We have a lot to learn and to understand.
Knowing the transient nature of probiotics in the gut, would it enhance having prebiotics always along with the probiotics? I think we are, sidetracking the prebiotics a bit, wherein it would be beneficial as science goes and what we have been reading.
Again, all probiotics are not the same, so depending on the probiotic, we may have different effects. But what we are observing right now with most of the probiotics which have been tested, that they are able to restore functions. So is that a drug effect? I don't know. And they are not able to cure disease, but they are helping our functions, our capacity to work at their best, at their optimal. And for me, it's like vitamins. If you want to have a proper functioning, you need nutrients and you need vitamins to have a proper functioning of the function. And vitamins are foods. So I guess that we are discovering that beyond nutrients, beyond micronutrients, we have also these probiotics which are providing something we were not aware of. But they are not forming a drug because they are participating to the normal functioning of our body.
Professor Bhattacharya, last question.
It's a nice presentation. I just would like to know whether you also try to identify the patrons, MAMPs, for example, because they can induce the expression of all these, human genes, important human genes, the components, cell surface components. So viability then will not be a strict requirement. Even the dead cells can also do the same thing. So did you try to work out that?
It's a good question. I have no answer for that. It's okay. Well, again, we are at the beginning of the understanding of the mechanism. So how far are they acting, how long, and, well, it's an open question. What I am conveying as a message that we have to take into account this cooperation. And we no longer can look at the gut without taking into account the microbiota which is there. And again, the microbiota, not who is there, but what functions are there. And in fact, we have maybe to think about the adequation of the right function of the microbiota with the right function of the guts. And this equilibrium between two functions, it's a new way of thinking.
A short question, please. Very short.
Just a comment. This is one of the most provoking talks in the last 2 days. If you look at it from a purely biological perspective, I think if I don't sound a bit iconoclastic, I think the language in which they communicate certainly is not the language of the central dogma which governs all processes in life. I think there is need for us to really look deeper into this, because this communication talk in relation to the processes in microbiota, at least some of which have extraordinarily different DNA modifying systems, makes us think that there are other things to look at here. I think there is need for us to really understand what that language is, even if the departure is from the central dogma for DNA, RNA, protein. I think that is what looking at.
Thank you for the comment.
Thank you for the comment.
Would you like to say anything?
No, that's okay. It was a very good comment.
I think on that note, we call a close to the session. We are going to have a good panel and you can have a lot of question answers then as well. And I would once again please give a round of applause to Professor Satoshi and Professor Zhang. Thank you very much.
Now I request Dr. Lahiri to please present the memento to our speakers. To Dr. Matsumoto. And Dr. Antoine. And now, may I request Gerard Denarius to please come up and give the memento to Dr. Lahiri. Oh, you closed it. Well, as usual, we are running late in time, and we have a small coffee break now, so maybe request you all to please collect your coffee, tea, whatever, and come in, and we could continue the session from here.